Hello everyone, welcome back to my YouTube channel. I know it's been a while since I've posted, but now we're kind of establishing a schedule where I'll upload one video every week. So, today's topic that we will be covering is cells. Now, what is a cell? A cell is the most basic unit of life. Okay, so cells can be classified into two types. The first type is prokaryotic, and this type does not have a fully formed nucleus or organelles. And the other type is eukaryotic, which has fully formed nucleus. So, an example of a prokaryotic cell, I'll just abbreviate it like that, could be bacteria. But eukaryotic cells actually include human cells or amoeba. Now that we've established the two types of cells, let's draw a diagram of both and just see what sort of organelles are present inside. Okay, so these are the three layers, and now inside we have DNA sort of shaped like this into a bundle with a protein we call histones. We have some ribosomes floating around. We have something similar to lysosomes but not exactly. And we have a flagellum over here, and we have these hair-like projections present on the surface called fimbriae. Now let's take a closer look at the bacterium. This is a capsule, and if we take a look at the capsule over here, we can basically just see that it's a bunch of monosaccharides bonded together and some peptides into the mix as well. This is basically how it looks. So, the next structural element of our bacteria is the cell wall. Now, the bacterial cell wall is a bit complicated, so I'll show you this after the cell membrane and the rest of the bacteria. The cell membrane is located here. This is a flagellum. These hair-like projections are known as fimbriae. This is our genetic material. These are just ribosomes. And these circles are what we call inclusion bodies. Now let's take a closer look at the cell wall. The cell wall of bacterium is divided into two types, gram-positive and gram-negative. The cell walls of bacterium are made up of a protein known as peptidoglycan, and they're made up of an acid known as teichoic acid. You can also find this in lipoteichoic acid form as well. So let's just draw what it looks like. So this is our gram-positive. These parts are just binds of proteins. This is our teichoic acid. Here's our peptidoglycan. And these are membrane proteins. As you can see here, the majority of the bacterial cell wall in gram-positive bacteria is made up of proteins, while the below part is made up of teichoic acid. This makes their cell walls relatively easier to break down compared to gram-negative bacteria. Let's take a look at that. Just remember, this is just a loose diagram. This is not an accurate representation by any means. So here at the top, we have these binds of sugars known as lipopolysaccharides. And this again is our teichoic acid. This is a membrane protein. And this is our protein structure. So here, basically, since the outside is made up of lipopolysaccharides and teichoic acid supports, this cell wall is much harder to break down than this, because proteins can easily be hydrolyzed. Remember this term, hydrolyzed. Basically, it means they can easily be broken down into long amino acid chains, into simple amino acid compounds. Now that's about the gram-positive and negative. Now we'll be seeing about the bacterial genetic material. So in the bacteria, genetic material looks a little bit like this. So I'll bind it together. Now this genetic material isn't free floating in the bacterium. There are some small proteins holding it together. These proteins we call histones. And another important thing to remember, the bacteria always will have a backup DNA here. Always. So these extra DNAs are known as plasmids. Let's take a closer look. So you can see here the main DNA is binded up with histones or histone-like structures and is responsible for cell instructions and cell function. But this other type of DNA, the plasmids, do not have histones. They are anhistonic and are responsible mainly for DNA repair or the transfer of genes from one bacterium to another. Let's see an example of this. So if we have one bacteria here, and we have another bacteria over here, the meeting point between these two bacteriums will be where the plasmid from the right moves to the left. Now let's say the right one is antibiotically resistant to a certain drug. Then the gene for that is in the plasmid will move over to the other bacteria and give it the same ability. Now let's look at the bacterial inclusion body. So, similar to how cells have lysosomes, bacteria have inclusion bodies. Lysosomes look like that with all of their proteins inside, but inclusion bodies, they're sort of very similar. They have the same enzymes and proteins present inside. Basically, they have the same function. They are just responsible for waste disposal. And you should know that lysosomes are actually part of the inclusion body family. It's just not very clearly mentioned what exactly are the inclusion bodies inside of bacteria, since there are many, many different types. Anyway, that's all for this video. I hope you liked this video, and if you liked it, please smash the like button down below and subscribe for more content like this. I couldn't fit eukaryotic cells into this video, so I will be making a part 2. Thank you for watching, and bye!